Over an average lifespan, a human male produces roughly 2 trillion sperm cells. A female, by contrast, releases approximately 400 mature eggs. Despite this staggering gap in volume, both biological systems share the exact same mechanical objective. They must reduce a standard diploid cell, containing a full set of 46 chromosomes, into a haploid gamete carrying exactly half that amount, utilizing the process of meiosis. This flowchart illustrates the dimorphic execution of gametogenesis. The male model is continuous and symmetrical, an unbroken production line churning out vast quantities of functional cells. The female timeline is discontinuous and asymmetrical, heavily gated by strict checkpoints to isolate a single massive cell. This pathway deliberately sacrifices volume to prioritize extreme cytoplasm retention and prolonged periods of meiotic arrest. Understanding human reproduction requires examining these two pathways as a dual evolutionary strategy. One optimizes for statistical probability. The other concentrates all available resources into a single provisioned payload. The male production engine comes online at puberty, localized entirely within the coiled seminiferous tubules of the testes. This cross-section shows the process initiating. A diploid spermatogonium clones itself via mitosis. One pushes inward, differentiating into a primary spermatocyte. Entering meiosis I, it divides into 2 haploid secondary spermatocytes. In meiosis II, they divide again, yielding 4 identical haploid spermatids. Technically, this specific point marks the end of spermatogenesis. The resulting spermatids are perfectly round and completely immobile. To become functional, these cells must enter a distinct secondary phase known as spermiogenesis. This is a physical transformation. The cell sheds excess cytoplasm, elongates, and develops a flagellum, morphing from a simple sphere into a streamlined spermatozoan. Finally, the mature sperm cell undergoes spermiation, detaching its head from the surrounding biological nutrient structures. Throughout this maturation sequence, developing sperm rely on specialized support structures called Sertoli cells, providing vital nourishment right until the moment of spermiation. Maintaining a symmetrical 1:4 division ratio, the male reproductive system acts as a relentless, continuous engine, securing a high daily yield of distinct motile cells ready for deployment. This exploded view breaks down the spermatozoan's payload: the head, neck, midpiece, and tail. The head is capped by the acrosome, carrying hydrolytic enzymes to penetrate an egg. Just behind, the midpiece packs a spiral of mitochondria, generating ATP to drive motility. Embedded securely within the neck is the proximal centriole, a critical piece of cellular hardware that will direct the first zygotic cleavage events post-fertilization. Generating hundreds of millions of these complex structures daily carries a massive energetic and hormonal cost. This circuit flowchart maps the neuroendocrine loop. The sequence fires when the hypothalamus releases GnRH to the anterior pituitary gland. The pituitary responds by releasing LH, which targets Leydig cells, and FSH, which binds to the Sertoli cells. When LH reaches Leydig cells, it stimulates systemic testosterone, driving spermatogenesis. To prevent hormonal overload, excess testosterone and inhibin travel back up the chain via a negative feedback loop, throttling pituitary and hypothalamus signals until the baseline is restored. This self-regulating autonomy allows male reproductive capability to operate as a closed-loop circuit, executing endless symmetrical divisions as long as the endocrine baseline is met. Pivot to the female system and the timeline shifts entirely. Here, the gametic clock initiates long before the female is even born. This timeline maps the fetal phase of oogenesis. In utero, millions of diploid oogonia differentiate and begin meiosis I, but abruptly halt. Arrested at prophase I, the genetic material is suspended in a holding pattern lasting over a decade. During this lengthy suspension, the vast majority of these cells undergo atresia. This massive biological attrition rapidly strips down the viable follicle pool before puberty. With the pubertal cycle's onset, surviving primary oocytes are triggered to resume meiosis I. But when division finishes, it is violently unequal. It produces one massive secondary oocyte and a tiny, useless first polar body. This extreme asymmetry forces the maximum possible amount of maternal cytoplasm and nutrients to remain hoarded inside a single surviving cell. This heavily provisioned secondary oocyte then advances directly into meiosis II. Yet it almost immediately collides with a second biological roadblock, halting its division again at metaphase II, just prior to ovulation. Oogenesis relies on decades of meiotic arrest, interrupted by asymmetrical divisions and severe attrition, to effectively engineer a single, highly concentrated payload. The arrested oocyte cannot survive a suspension lasting several decades entirely on its own. It requires complex physical armor. This diagram illustrates folliculogenesis, the physical construction of a biological housing. It begins when granulosa cells wrap the oocyte, forming a primary follicle. As it transitions to a secondary follicle, theca cells add structural density before demanding a massive structural shift. A distinct fluid-filled cavity known as the antrum swells within the cellular layers, preparing the entire complex for its eventual pre-ovulatory state. Simultaneously, the outer layers differentiate into a highly cellular theca interna and a fibrous theca externa to finalize the architecture. While the gamete's genetic division remains deeply paused, its physical housing is dynamic, constantly evolving to protect the heavy cellular investment inside. These side-by-side equations quantify the stark divergence in meiotic efficiency. In a male, 25 meiotic cycles yield exactly 100 spermatozoa. In a female, 100 meiotic cycles are required to produce 100 ova. The male system guarantees a fully completed, independent gamete at the end of every cycle, regardless of its future utility. The female gamete, however, is ovulated in an entirely arrested, incomplete state. That secondary oocyte, halted at metaphase II, lacks the biological capability to complete meiosis II on its own. The final division, releasing the second polar body to create a true ovum, is exclusively triggered by the physical fusion of a sperm cell. While male gametogenesis is an autonomous, self-contained engine, the female gametic journey is uniquely relational. It remains biologically unfinished until the precise moment of fertilization.